Hello friends, welcome to my course on Microbial Biotechnology. We are in module 3, which is dealing with the physiology of microorganisms and adaptation to environment. In this lecture we will be discussing about the physiology of extremophiles and their adaptation, and then we will be discussing certain concepts like energy storage, temperature, pH and pressure conditions under which these extremophiles survive. So, briefly we will be discussing about energy storage, as I told you, types of the different extreme environments microorganisms live in or face, an effect of environmental factors on microbial growth, adaptation to variable temperature, variable pH and variable pressure. Let us start with the energy storage strategy and adaptation mechanisms in microorganisms. Humans, particularly those living in very difficult environments like the high Himalayas or other areas where there is only seasonal supply of food materials, are long known to store food to cope with the scarcity of food in the lean season. And this is a overall strategy actually employed by many higher organisms. Microorganisms also have evolved mechanisms to store energy rich compounds during periods of nutrient abundance, allowing them to survive times of nutrient scarcity. These storage strategies highlight the metabolic flexibility and resilience of microorganisms, enabling them to thrive in diverse and fluctuating environments. Microorganisms synthesize and accumulate a variety of storage compounds that serve as carbon, energy and phosphate sources. Some of these key storage compounds we will be discussing in this lecture today, like storage carbohydrates, energy rich lipids, inorganic reserves and several others like trehalose. Let's start with the storage carbohydrates, how microbes store these for their future use. Let's start with glycogen, which is a polysaccharide made of glucose units. It serves as a primary carbon and energy reserve in many bacteria, fungi and even in animals. When nutrients are scarce, glycogen is broken down to release glucose for energy production. Then there is starch, which is primarily found in plants and some algae. Starch is a polysaccharide composed of glucose units, and it serves as a long-term energy reserve and can be hydrolyzed into glucose when the need comes. Then there are energy-rich lipids like polybeta hydroxybutyrate or PHB, which is a biopolymer the bacteria store. PHB is an important carbon and energy reserve. It is particularly prevalent in bacteria like Rhodovector and Bacillus, but it is not found in eukaryotes. It serves as a reservoir for carbon, especially when external carbon sources are limited. Then we have neutral lipids in filamentous fungi and yeast. They often store triglycerides within their vacuoles. These neutral lipids provide an energy source that can be mobilized during nutrient shortages or environmental stress. Then microbes also store inorganic material or inorganic compounds. For example, volutin granules, these are the phosphate granules. These are found in both prokaryotes and eukaryotes. Volutin granules are composed of polyphosphates and serve as a phosphate and energy reserve. These reserves are crucial for essential metabolic processes like nucleotide synthesis, cell division and ATP generation. Then we have sulfur granules in sulfur bacteria like Thiobacillus. They store sulfur granules by oxidizing hydrogen sulfide. When external sulfur sources are exhausted, these granules are oxidized to sulfate, providing reducing equivalents for critical metabolic processes such as carbon dioxide fixation and oxidative phosphorylation. Then there are other compounds like trehalose, which is a disaccharide synthesized by filamentous fungi, yeast and some bacteria. It plays a key role as a protective agent during environmental stress conditions like desiccation, heat shock or freezing. Trehalose stabilizes cellular structure and prevents damage by forming protective glasses around proteins and membranes, and it's a very important molecule for survival under harsh conditions. Hydrolysis of these storage reserves, how these stored compounds are finally utilized in the time of need. The breakdown of these energy reserves is facilitated by enzymes like phosphorylases, which catalyze the hydrolysis of polysaccharides like glycogen and starch to release glucose 1-phosphate. This glucose can then enter various catabolic pathways such as glycolysis or the pentose phosphate pathway to produce ATP and support the microorganisms' energy needs during the period of nutrient scarcity. Now, let us look into details of survival of microorganisms in extreme environment. Extreme environments like acidic or hot springs, saline and alkaline lakes, deserts and ocean beds are there in nature, and they are inhospitable habitats for conventional life forms like us or even plants. These environments can be characterized as conditions that surpass the normal acceptable range, thus qualifying as extreme conditions. Remarkably, certain microbes are capable of thriving and proliferating in such harsh settings. These organisms not only endure specific extreme conditions, but often depend on them for their survival and growth. Extremophiles, as they are called, which survive in such harsh settings, are predominantly found within the microbial realm, demonstrating a remarkable ability to tolerate a wide range of environmental extremes, surpassing that of other life forms. So, these are environmental niches in which other organisms can barely survive. So, as already discussed, when we tell extreme environment, it is not just of one type. The sea ice, permafrost and polar region, coal seeps and the mud volcanoes, shallow water hydrothermal vents, hot springs, and then hyperacidic lakes and volcanoes, deserts and arid environments with almost nil water, and then the acid mine drainage, and then the deep sea anoxic lakes, deep sea hydrothermal vents, then serpentinizing environments, then deep sea sediment entrances, marine and continental subsurfaces. We have soda lakes and hypersaline lakes, then nuclear contaminated sites which may be natural or man-made. Then we have ophiolites and continental serpentinization. So, these are the types of extreme environments where we can find various microbes surviving by adaptations. So, these kind of microbes we call as extremophiles because they can survive in extreme environments, can be simply named as acidophiles, alkaliphiles, piezophiles, halophiles, gerophiles, radiophiles, metallophiles, thermophiles and psychrophiles. We will be discussing about some of these today and in the next lecture. So, how does environmental factors affect microbial growth is an important point to ponder before we go for the detailed discussion. So, we have seen that microorganisms can withstand and survive in extreme environmental conditions. They are the only ones which can be present in such habitats which are otherwise inhospitable to other organisms, and for them this is a normal habitat. But for these, the microbes need to adapt and respond to changes within their habitats to survive. Ecological distribution and understanding the impact of the environmental microbes will help us comprehend the survival strategies in such environmental extreme environmental conditions. So, adaptations to various conditions. So, a microbe may be under various kind of stress, environmental stress, due to which the DNA may be damaged, the protein may be damaged, and then the reactive oxygen species effect will be there. And then how they adapt to these kind of environmental challenges is what we are trying to understand in this lecture. So, for this they have evolved various mechanisms to manage the specific stresses they encounter. There is not just only one kind of challenge, and so there are also similarly multiple kind of strategies to manage these kind of stresses. The microorganisms adjust their biochemical pathways and structural components to maintain functionality and stability under extreme conditions such as high temperature, pressure or salinity. So, For example, if such stresses cause DNA damage or it causes protein damage, the microorganism will try to evolve mechanisms which will safeguard the DNA, which will safeguard the protein. So, such adaptation involves changes in the enzyme activity, membrane composition and osmotic regulation to ensure survival and proper cellular function in harsh conditions. Adaptation to variable temperature. Earth's surface temperature ranges from almost minus 100 degree in Antarctica to 495 degrees centigrade in the deep sea hydrothermal vents. In geothermal or magmatic environments, temperature can exceed 100 degree due to high pressure. The highest surface temperature recorded is about 71 degree centigrade in Iran's Lut desert, that is on the surface. Microbial life can survive from minus 25 to 130 degrees centigrade with metabolic activity ranging between minus 20 to 122 degree centigrade as reported on today. Now, what is the effect of temperature? So, if you look the growth rate as the temperature increases, the growth will be more and then reach an optimum value and suddenly it will start declining. So, temperature greatly affects microorganisms as they lack the ability to control their internal temperature. Enzyme catalyzed reactions essential for growth are particularly influenced by temperature. Each enzyme has an optimal temperature at which its function is best or optimum. So, cold environments or the psychrophiles. More than 80% of Earth's biosphere is permanently below 5 degree centigrade and diverse microorganisms have been discovered in these seemingly inhospitable regions worldwide. The ability of organisms to survive at low temperature is determined by the freezing point of water inside the cells. Crystallization of the intracellular water is lethal for most organisms. It will rupture the cell except for the nematode Panaglomys davidi, which can survive the freezing of all the body water. Psychrophilic species such as Swanella, Vibrio, Micrococcus, Methanogenium and Bacillus have been identified. The genus Moritella consists exclusively of psychrophiles. Archaea, cold living organisms that inhabit temperatures below 15 degrees centigrade had adapted to the challenges of low temperature and high viscosity in aqueous environment. So, these are some of the examples of psychrophiles. You can see the snow surface with snow algae here in this picture and the lichen here, Xanthoria. These continue to photosynthesize at minus 24 degree centigrade. So, how do psychrophiles adapt? So, there are various mechanisms like at the genetic level there may be horizontal gene transfer, acquiring genes, fitness genes which help them in adapting to such conditions. Then there is a genetic capacity to store energy in molecules like triacyl glycerols and polyhydroxyl alkanoate, which helps meeting its energy demands at a later point of time. Then there is a regulation of the membrane fluidity which will not allow entry and exit of metabolites, rather control it, regulate it. And then other strategies include amino acid substitutions like one case is over representation of glutamine, threonine and hydrophobic amino acid residues. Then there is increased protein structural flexibility, and very interestingly they have antifreeze and ice binding proteins which help in protecting the organism as a whole, and then also the deployment of compatible solutes and polyhydroxylated soluble compounds like glycerin, betaine, glycerol and trehalose. So, some of the common structural modification of psychrophilic enzymes result in decreased thermostability, increased flexibility and increased specific activity. So, these increased flexibility, decreased thermostability and increased activity. So, here the lysine arginine ratios are increased, arginine proline isoproline content are reduced, and then there is a glycine clustering at the catalytic site, a decreased interdomain and subunit ion electrostatic and aromatic interactions happen. Increased hydrophobic surface residues, reduced core hydrophobic residues, then increased size and number of enzyme cavities happen, and extended surface loops, predominance of neutral amino acids, reduced proline content, and increased mobility of the catalytic site. So, these are some of the common structural modifications of enzymes in the psychrophiles, which help in increase flexibility, decrease thermostability, and increase specific activity. Now, let us look into the other extreme, warm temperatures. Such organisms which survive in higher temperatures are known as thermophiles. Around 25 to 40 degrees centigrade they survive and exhibit growth rates temperature exceeding 45 degrees centigrade. Various extreme environments, both terrestrial and submarine, harbor high temperature conditions suitable for the growth of thermophiles. These habitats encompass thermal vents, hot springs, and boiling steam vents, while surface soil and compost piles also provide suitable environments. Thermophiles can be categorized into two groups: facultative thermophiles, which are capable of thriving at both high and moderate temperatures, and obligate thermophiles, which require high temperatures for growth. So, some of the examples of thermophiles are Methanosarcinia thermophila, then Methanobacterium wolfii, Methanobacterium thermoautotropicum, then Archoglobulus profundus, then Alicyclobacillus acidoterrestris, and Acidocaidarius. How adaptations in higher temperature happen? So, here one of the strategy employed is having smaller genome size, then genes for polyamine biosynthesis, pre-A helicase, DNA binding protein HU, heat soak proteins, GRPE, DNAK, DNAJ, GROW-EL, then co-chaperones, GROW-ES, heat inducible transcription repressors, and cold soak proteins from the CSP family are deployed. So, this helps in the adaptation to higher temperature. Then there is preferences for G and A in the first and third codon positions, and overrepresentative amino acids like I, V, Y, W, R, E, L. Then presence of diverse insertion sequence elements. Pentose phosphate pathway favored at high temperatures over glycolysis. Glycosylation of carotenoids and thermophilic aids in thermophilic adaptation. Then there is the absence of the uracil DNA glycosylase gene, which is linked to cytosine deamination. Heat soak proteins. Thermophiles produce HSPs or heat soak proteins and chaperones that assist in protein folding, especially under high temperature stress. HSPs are activated in response to heat soak and are classified based on their molecular mass, example HSP 70, HSP 60. These chaperones help fold nascent or unfolded polypeptides and prevent protein aggregation. The well studied HSP 60 chaperonin complex or the GROW-EL, GROW-ES in bacteria plays a key role in protein folding by creating a protected environment for proteins to fold properly. If proteins are unable to fold, proteosomes, not thermosomes, target these misfolded proteins for degradation, ensuring cellular homeostasis. So, such proteins will be immediately got rid of. So, here we can see there is some stress which induces the HSP 70, 90 chaperone signaling, or and then it will help in the synthesis of HSP chaperones and other stress proteins. Then there is this HSP 70 stabilizing and preventing the aggregation. So, this protein without this will be denatured. So, this is the aggregate, and then also 60, 70, 80, 90 will help in the refolding as discussed in the earlier slide. Thermostable proteins. Proteomic studies have revealed that protein stability in thermophiles is vital for adapting to high temperatures, especially for regulatory and catalytic proteins. Thermophiles have more disulfide bonds than mesophiles, enhancing protein stability. However, the intracellular reducing environment complicates disulfide bone formation. Genomic data suggests thermophiles possess disulfide oxydoreductase proteins which aid in stabilizing proteins through disulfide bond formation. Thermophilic enzymes possess unique architectural properties which contribute to their thermophilicity. There is a high occurrence of arginine, aspartate and glutamate amino acids, abundance of salt bridges, then there is a compact hydrophobic core, then numerous surface ion pairs, and there is additional hydrogen bonding and shorter loops. Let us now discuss about the survival of microorganisms under extreme pH condition, and what is a favorable pH growth range and what is the optimum pH favored by certain organisms. So, if you look into this figure, as we go from the acidic to the alkaline through the neutral range at 7. So, here we see the pH range of certain body fluids or biological fluids. So, the pH of the human stomach fluid is somewhere here between 1 and 2, lemon juice will be somewhere around 2, grapefruit will be 3, oranges, beer will be something here, tomato juice, cheese, and then some meat products, milk will be somewhere near neutral, and pure water of course is the most neutral thing, and that is the standard from where we measure pH. And then human blood little bit slightly towards alkaline, seawater, baking soda, soap, household ammonia, bleach. So, this is the scale from 1 to 14, and this is the material list which gives an idea about pH, which material is having how much of a pH. And then here are the pH optima of some of the microbes corresponding to these. So, you have ferroplasma species which is surviving at very low temperature between 0 and 1. Then you have Bacillus alkalophilus which is surviving something around 11. Then you have various others like Lactosomonas alkaline, and then you have Lactobacillus acidophilus, slightly acidic conditions. So, these gives us an idea that microbes can survive from almost 0 pH to something around 11 to 12 pH, and there is a whole list of microbes over here, and some examples are given over here. So, how do they survive in such extreme pH conditions of acidity and alkaline conditions? So, microbes have evolved to thrive in a wide range of these pH conditions, and their growth is often optimized for specific pH ranges. Each species has a specific pH growth range and optimum. So, those who survive in the acidic range are called as acidophiles. They grow optimally between pH 0 and 5.5, thriving in highly acidic environments like acidic lakes, hydrothermal systems, animal stomachs. Then there are the neutrophiles who grow between roughly 5.5 to 8. Then there are alkaliphiles grow optimally between 8 to 11.5, with some species thriving in even more alkaline conditions, 9 to 11, 13. Alkaliphiles are divided into two. Obligate alkaliphiles grow only above pH 9. Then there is a facultative alkaliphiles can survive in both neutral and alkaline conditions. So, we have already given examples of the acidophiles and alkaliphiles in the earlier figure. Still, as the name suggests, Acetobacter aceti, these are alkaliphiles, and then you have Mucoracimosus, and then you have those are acidophiles, and then you have alkaliphiles, the Anabena species, and Microcystis species, which are basically Bruggen algae species. How do these microbes adapt to low pH, or how do acidophiles survive in low pH? So, They do it by various mechanisms or due to various features they have. For example, they maintain pH homeostasis through impermeable membranes, proton transporters, cytoplasmic buffering, potassium ions, and proton extrusion. Then they also adapt metabolic activities or metabolic adaptations are there. They have limited gene pools for carbohydrate, amino acid, and fatty acid metabolism, relying on enzymes like sulfur and sulfite reductase for energy production processes. And then their genomic features are something unique. They have smaller genomes with 600 to 1200 genes lost, including those for cofactor biosynthesis and sporulation, and contain many pseudogenes. This kind of a maybe backward evolution or devolution. Then protein and enzyme adaptations. Acidophiles have more acidic amino acids, fewer salt bridges, and enzymes like glutamate decarboxylase for acidic conditions. Then they have transporter systems. The NHAD transporter helps maintain pH homeostasis across a range of conditions. So, these are the various adaptations strategies adopted by acidophiles. Then also their counterparts, the alkaliphiles, have similar or some specific adaptation mechanisms. For example, the alkaliphiles generate ATP at high pH by producing carotenoids that help pump protons near ATP synthesis and possesses cytochrome C 553 and ATP synthase genes. Then they have their membrane adaptations, which contain cardiolipin, which retains protons near the membrane to prevent diffusion into the alkaline environment. Then there are certain genomic features. Alkaliphiles have larger genomes compared to acidophiles, with expanded carbohydrate and amino acid metabolism, along with extra chromosomal elements and insertion sequences for high pH adaptation. And as in the last case, they have transporter systems, HAP and HAFA transporters. They counter high salinity and alkalinity, and there is a HAM-RP which helps maintain homeostasis in high pH. So, these are the various transporter systems, as you can see, which helps maintain homeostasis in alkaline pH. Let us now discuss about adaptation of microbes to high pressure or variable pressure. For example, they are found in deep sea environments. These are the hyperpiezophile. In the deep sea ocean, pressure can reach over 100 megapascals, approximately 1000 times atmosphere times of the atmospheric pressure. These conditions impact water's physical and chemical properties, affecting microbial life. So, there is subsurface environments, deep geological formations such as deep mines or hydrothermal vents also experience a high pressure. And then there are low pressure areas, or these organisms, they survive there. These are known as hypopiezophiles. For example, in high altitude environments in the mountains like Everest, at high altitudes atmospheric pressure will drop significantly. At the summit of Mount Everest, which is around 8,900 meters above sea level, the atmospheric pressure is only about 1/3 of that at the sea level. And then space and high altitude atmosphere, the vacuum of space and the near vacuum conditions in high altitude environments present extreme low pressure conditions that can challenge microbial survival. So, this is a graph of optimal growth, temperature and pressure for some piezophilic bacteria, blue, and then archaea, which are red, and as you can see, their preferences are very distinct. So, they can survive at say 10 degrees centigrade and up to 120 megapascals, and here these are surviving around 110 degree centigrade under something 20 megapascals. So, what are the effects of high pressure on the cellular components? So, high hydrostatic pressure affects several components inside the cell. For example, under high pressure, the lipids in the membranes will be affected. The lipids are fluidic, as we know, in the membrane, but due to increased pressure, they may become compacted and rigid. So, high pressure increases rigidity, reducing fluidity and affecting the membrane protein function. Then in the case of multimeric proteins, high pressure may disrupt protein subunit assembly and protein-protein interactions. So, they will be disrupted and they won't be coming together. So, they will become non-functional. Then protein structure also can be affected. This is a native protein. Due to high pressure, the protein becomes unfolded. So, it can denature proteins, causing misfolding and aggregation. And then the cellular motility is affected. Pressure impairs the flagellar function. The flagella won't be able to function properly, and even within the cell, the cytoskeleton dynamics and ATPase activity will all be affected, and this will result in the movement and also the cell division. So, due to a mobile component, due to high pressure will become non-mobile. Then finally, high pressure affects protein translation, which is very interesting. It slows translation. So, this is high pressure. So, this is normal translation happening. Due to high pressure, there will be no translation. So, how does it happen? It destabilizes ribosomes and translation factors, reducing protein synthesis efficiency. Probably as these multimeric proteins are not allowed to come together or they are disrupted. Similarly, the ribosome machinery will also be affected, thereby stopping protein translation. So, we can see the effect of pressure on the cellular components are very, very diverse. Whatever, it actually restricts the growth, or it's almost makes life impossible for microbes. So, how do these microbes adapt to such high pressure or low pressure environments? So, let us discuss with the hyperpiezophile first. They thrive under extremely high pressure conditions, such as those found in the deep sea environments, and some of their adaptation mechanisms include membrane adaptations. So, as the membrane is disrupted, we have discussed. So, high pressure resistant membranes, they have evolved. These organisms often have membranes rich in saturated fatty acids, which help maintain fluidity and functionality under high pressure. Then there are specialized lipids. Some produce or modify lipids to stabilize membrane structures under high pressure. Then there are protein adaptations. As we have seen, the proteins are either unfolded or there is disruption in the multimers. So, here pressure stable proteins are being evolved or adapted, and they are stable and functional under high pressure, often through more compact structures. Then there are evolution of separin proteins. They produce separin proteins that assist in maintaining the proper protein folding. Adaptations to low pressure environments, hypopiezophile. So, this is a new area of research with implications for predicting microbial activity in cloud and bulk atmosphere on Earth, and for modeling the forward contamination of planetary surfaces like Mars. So, here also there is a pressure responsive stress proteins. These organisms produce general stress proteins, do not specifically for high pressure. Membrane adaptations like reduced membrane fluidity, they may not have specialized adaptation to increase membrane fluidity under high pressure. Low pressure enzyme efficiency, enzymes in these organisms are optimized for normal pressure and may not perform well under high pressure. Then there is a habitat selection. They avoid high pressure environments by inhibiting areas with normal or low pressures. So, with this we come to end of this lecture on extremophiles. Thank you for your patient hearing.